Hey everybody, so we're going to keep talking about cellular respiration today, and we're going to move on to the Krebs cycle and talk a little bit about electron transport as well. So the Krebs cycle is also called citric acid cycle, or sometimes they'll see it as the tricarboxylic acid cycle or TCA cycle. They all mean the same thing. And the function of this is to further oxidize pyruvic acid or pyruvate to carbon dioxide. So we talked in class about how glycolysis is the partial oxidation of glucose. So the Krebs cycle is going to be where you further oxidize that pyruvic acid. You're going to produce your carbon dioxide in this step. You're also going to make more of those electron carriers, so you're going to have more NAD plus converted to NADH, and now we are looking at a different one. FAD is going to be made into FADH2, and this is all happening in the matrix of the mitochondria. So here is the cytoplasm of the cell. Here's your pyruvate, which is produced during glycolysis. Keep in mind that you have two of these produced during glycolysis. They will be moved into the mitochondria through a protein in the mitochondrial membrane. You're going to release a carbon dioxide. You're going to have an NADH formed. Coenzyme A is going to come in, and you're going to make something called acetyl coenzyme A. And that acetyl coenzyme A is going to be really important for bringing this molecule into the Krebs cycle. So things you need for Krebs cycle: your pyruvic acid. So you're going to do this with one pyruvic acid at a time from glycolysis. So for every one pyruvic acid, you're going to have one coenzyme A, 4 NAD pluses, 1 ADP, and 1 FADH. Now, keep in mind that this is for one pyruvic acid, and you produce 2 pyruvic acids at the end of glycolysis. So really, you need to double this if you're asked how many molecules are required for Krebs cycle after a round of glycolysis, because you're producing 2 pyruvic acids for every glucose, so you have to do this twice for every 1 glucose. Then your products are going to be 3 carbon dioxides, acetyl coenzyme A, 4 NADHs, 1 ATP, 1 FADH2. Again, same situation. This is for every 1 pyruvic acid that you break down, but again, you're getting 2 pyruvic acids for every glucose. All right, and I do want you to see that you've got your requirements and then your products, so they are very similar. So you start with pyruvic acid, but you break it down into 3 carbon dioxides. You start with coenzyme A, it becomes acetyl coenzyme A. You start with 4 NAD pluses, it becomes 4 NADHs. So you guys get the idea, and again, you have to double the list for every glucose because this list is based on every 1 pyruvic acid. So here's just a quick breakdown. So again, this is in the mitochondria. You have your pyruvate, which is going to be brought into the mitochondria. Remember, it was formed in the cytoplasm. You're going to release a carbon dioxide. You're going to form an NADH. Coenzyme A is going to come in, and it's going to form this compound acetyl coenzyme A. Once you have that, then you're going to start Krebs cycle. You're going to release that coenzyme A again. And as you go through Krebs cycle, 2 more carbon dioxides are released. You have 3 more NADHs formed, 1 ATP formed, and then FADH2 formed. All right, and so this is going to happen once for a pyruvate, but again, 2 pyruvates from glycolysis, so twice for every glucose that you're breaking down. So the Krebs cycle produces most of the cell's energy in the form of NADH and FADH2. Those NADH and FADH2 molecules are then going to get shuttled somewhere else where they are used to produce ATP. So Krebs cycle does not require oxygen. However, oxygen is required at the next step where you're going to use these electron carriers and drop off electrons. Just a quick comment that the ATPs that are produced in Krebs cycle and glycolysis are through substrate level phosphorylation, and that is what we talked about that last time. That's when you have that inorganic phosphate group transferred from a substrate to an ADP to make ATP. And so you have an enzyme that is helping to facilitate that. All right, the next step is electron transport chain. Sometimes you'll see an electron transport system. In this class, I tend to use ETC, or electron transport chain. And this is really a collection of proteins that are linked together in units. Now, a lot of times I talk about this sort of like playing hot potato, that these proteins are passing these electrons down, sort of like you would pass the hot potato down the line when you're playing that game. So the electron transport chain uses a set of cytochromes. These are going to be iron-containing proteins that pass electrons. And these cytochromes alternate between the reduced and oxidized forms, and they pass those electrons down to an oxygen. So the function of this is to convert the NADH and FADH2 to use the electrons that they're carrying and the energy that they have. They're using that to make ATP. And this is happening in the mitochondria Christi. Things that you need, the requirements for electron transport chain. You need to have your electron carriers there to drop off the electrons. So you've got your NADH and your FADH2. You need to have some ADP so that you can make ATP, and then you also need your oxygen. So we say that cellular respiration is aerobic, or it needs oxygen, and this is the step that needs the oxygen. And so when you look at the overall equation for cellular respiration, you see the 6O2. This is where those 6 oxygen molecules are used. Now the products, as things that are produced, you go back to NAD plus and FAD after these molecules drop off their electrons. The ADP is converted into ATP, and then this is also where your water is produced. So again, if you look back at that equation for cellular respiration, you see that your products include 6 carbon dioxides, which were produced during Krebs cycle, but also 6 waters. And so this is where the 6 waters are produced in cellular respiration. Now, what do we get out of this in terms of energy? We keep talking about how we're getting all of this energy out of cellular respiration. You're getting a lot of that here in this electron transport chain. So for every NADH, you're going to get 3 ATP molecules produced, and then for every FADH2, you will get 2 ATPs. And we're making these ATPs in a slightly different way. This is not substrate level phosphorylation. This is oxidative phosphorylation. And so we're going to talk very quickly about the chemiosmotic hypothesis. And this says that this electron transport energy is used to move hydrogen ions or protons across the Christi membrane, or that inner mitochondrial membrane. And then ATP is generated as those ions diffuse back into the matrix. So let's look at a diagram of this really quick. So just to kind of summarize where we're at in the cell, this down here is a mitochondria. So you have your outer mitochondrial membrane, then your inner mitochondrial membrane. The innermost space is the matrix, so this is where Krebs cycle was taking place. And here you have your intermembrane space. And then on the outside, again, that would be the cytoplasm. So glycolysis would happen here. The pyruvate would travel into the mitochondria. You have Krebs cycle take place inside the matrix. And now you've got all these electron carriers that are going to carry the electrons to this electron transport chain system, which is located in the inner mitochondrial membrane. Here we've just zoomed in on that membrane. Okay, so this is the inner mitochondrial membrane. It is a bilayer membrane, as you can see. You have your proteins. So these are like the little bluish green guys in here. All right, and so these NADH molecules are going to come in. They're going to drop off their electrons. As they do that, they're going to go back to that NAD plus form. The electrons are going to get passed from one protein to another down the line. As the electrons get passed, hydrogen ions are going to get pumped across this membrane through these protein channels. Okay, now what you're doing is you're pumping them into this area here in between the two membranes, that intermembrane space. So it's a really small space. You're pumping lots of hydrogen ions into that space. And so what's going to happen is the hydrogen ion concentration is going to build up on this side. Now what we remember about diffusion is that if you have a really high concentration on one side of a membrane and then a low concentration on the other side of the membrane, those hydrogen ions are going to want to get back through to the other side. So once we pump all of these hydrogen ions up here, they are going to diffuse back. And they're going to move back through one specific protein called ATP synthase. Okay, and this ATP synthase is what is going to generate this ATP. And so as the hydrogen ions move through ATP synthase, it is going to form, this molecule is actually going to spin, this protein is going to spin, it's going to make ADP into ATP, and this is the oxidative phosphorylation. So this is where your ATP is being made, okay? Other things that are happening here, as these electrons travel through the electron transport chain, right here we have the formation of water. So at the end of this chain, you'll have some hydrogen ions, some oxygen, grab onto some electrons, and together they will form water. So this is where the water is produced during cellular respiration. It's produced at the end of the electron transport chain. All right, so I know that was a lot, but essentially you really do need to know ATP synthase. So it's got that ASE on there, all right, so it's helping to build something. ATP synthase, it's building ATP, it's synthesizing ATP, okay? So it uses the flow of hydrogen ions to make ATP. It works like an ion pump in reverse, or like a water wheel under the flow of hydrogen ions, kind of like water. And so essentially what happens is, as these hydrogen ions move through the proton channel, this is going to spin. So you can see the fancy little arrow there. As this spins, as the hydrogen is pumped back into the matrix, the ADP and inorganic phosphate are bound together and it makes ATP. So this is oxidative phosphorylation at the end of the electron transport chain, and this is where we're going to get lots of this ATP generated as the end result of cellular respiration. So, your lecture question for today: can you relate how ATP synthase works to what we learned about energy? So how is energy being converted, and why is this necessary for cells? So when we talked about energy, we talked about the different forms of energy. We talked about potential energy, chemical energy, kinetic energy, so that energy of movement. So I want you guys to think about how ATP synthase works. I talked about how it's spinning and how that is part of how ATP is made. So I want you guys to think about how energy is being converted, and again, why this is necessary for cells. All right, thanks guys.